So the term microscopic means that any microorganism which are very small and can be seen only with the aid or use of a microscope. So this diagram shows how basically this culturing is done. So we need to use an instrument called Petri dish, where we put agar jelly with growing media, because just agar is not enough. Agar is a solidifying agent to make a jelly, and to that jelly we have to include all the required nutrients by the microorganisms. So when we make this agar, we add a lot of nutrients, and then when it is solidified, it's like serve as a jelly where the bacteria or any other organism can grow. So we using an inoculating loop, we introduce the pathogen or the microorganism into the media without touching or without letting other organisms to enter. So that is a laboratory procedure. So in coming lessons, we will explain you how this process can be done without introducing other organisms into the this highly rich nutritious medium. Then only we can separate the organism that we are interested. So these are some images to show you and to be familiarized with how they look like. This is a bacterial culture where you can see different colored bacteria, and the main way to identify bacteria are they form very bright, shiny, circular colonies most of the time, and you can see they appear in various colors. So all these are bacteria. They look very shiny. And when it comes to fungi, this is a fungi growing in artificial media. Again, the fungi is introduced and isolated in a separate nutrient medium where you can see the fungi is growing. So this is fungal colony where you can see like cotton-like. So fungal colonies look like cotton, not like bacteria. And this is a virus multiplication, because unlike bacteria or fungi, we cannot grow viruses, because they are obligate parasites. They cannot live in other media. They need a living cell. So in order to get the viruses, what we have to do is we have to first grow some cells, animal cells or plant cells in a culture, and then we introduce the virus where we can extract virus particles when they grow inside the cells. So that is how we harvest viral tissues or sorry, we can't call it viral tissues, virus particles. So that is why I mentioned here, viruses are produced, or it's just a matter of multiplication in cell cultures in artificial media. So again, there are a few terms that you have to be familiarized with. Pathogenicity. Pathogenicity is the ability of a pathogen to cause a disease on host plant. So what is a host? In pathology, host is an organism that harbors or retains the parasite or the pathogen. And susceptibility. A host plant which is not have the ability to resist the pathogen attack is called a susceptible host. Then the host is considered resistant when it has the ability to exclude the pathogen. So we can keep away the pathogen without getting the disease. But there is another term called tolerance, where it is not resistance, but tolerance is the ability of a plant to be colonized by a pathogen. Pathogen comes into the plant, they get colonized, but without dying or demonstrating disease symptoms, the plant can withstand it. So that is called tolerance. So the first two are genetically controlled because they have respective genes to maintain the susceptibility and resistance. However, tolerance is not controlled by the genes. It's the inherent ability or the strength of the plant that can tolerate the disease. So this is the last slide. I will just go through it and then you can later refer to these terms because these are really important. A parasite is an organism which lives on or in another organism and obtain its nutrients from there. An obligate parasite is an organism which is wholly dependent on its nutrients on another living entity. Obligate parasites are biotrophs, which are also depend entirely on living host for its nutrition. An autotroph is a plant that can make its own food through photosynthesis. A facultative parasite has the ability to adapt to an alternative model of living, that is parasite or saprophyte. Saprophytes are the organisms that gain their nourishment by digesting dead organic matter. So I copied this text in order to give you some terminology and to be familiarized with. So in addition to that, there are various terms that you come across during this semester in plant pathology course. So I would suggest you to keep a vocabulary of technical terms which will help you in better understanding of the course. So I expect you to make a list of technical terms that are taught in the class and find the meaning of each of these terms. So this is the end of this session. So we will get on with the regular lectures from the next session. Thank you.